Please appreciate this presentation has been prepared with all sincerity and to the best of knowledge of students, teachers, and volunteers. We welcome words of encouragement and also suggestions for improvement. For use in academics or any kind of exams, please verify and correlate with your course books. For becoming an associate or sending contributions in different subjects including storytelling, please get in touch with your brief profile at baskenjoy@gmail.com. So hello everyone. Today we'll be taking a look at the first chapter in class 9 biology, which is the fundamental unit of life, and this is lesson number 1. So the discovery. So Robert Hooke discovered cells in the year 1665 with the help of a self-made primitive microscope. So this image over here on the left shows the microscope used by Robert Hooke to find out or discover cells. And this microscope over here in the right is the new modern compound microscope. With the help of this, we can dive even deeper into the world of cells, and these days we even have electron microscopes through which we can study the composition of these cells. So next up, we'll look at the division of labor in living things. So in humans, we have the heart to pump blood, the stomach to digest food, we have the lungs to breathe air, etc. Similarly, there's a division of labor found in cells. And like we have the heart, stomach, and lungs in humans, each cell has their individual cell organelles, which have their own functions, which help the cell to basically live. So some examples are the mitochondria, nucleus, etc. And we'll get into them later on. So now we'll look at the main components of a cell. So the first one is the plasma membrane. It is the outermost layer of a cell which covers it like a membrane, or in other words, it encapsulates the cell. So as you can see here, this blue arrow is pointing at this plasma membrane. So as you can see, this is a darkish pink color. It's surrounding the cell. It's basically covering the cell. It's like a wall. You can think of it in a way. So next up, we'll look at the diffusion of gases through the plasma membrane. So in general, diffusion is the movement of particles from a region of high concentration to a region of low concentration. It is through diffusion that the cell is able to gain oxygen and remove carbon dioxide as waste. So now this picture over here clearly shows what I just said. So this is a normal animal cell, and if this is the surrounding medium of the cell, then as you can see, if oxygen is present in high quantity outside the cell and it's present in low quantity inside the cell, what the oxygen will do, it's going to move into the cell via diffusion, and CO2, which is generated in the cell by the process of digesting food, it's going to exit the cell. So there's high concentration of CO2 inside the cell, low concentration of CO2 outside the cell, so it's going to exit the cell via diffusion. So this short video clip shows diffusion in lung cells. So what lung cells are a bit different, as actually oxygen is exiting the cell. So this over here is a capillary. It's basically one cell thick, and what you can see here are the red blood cells moving across here. So what lung cells do, as we breathe in air, the oxygen rich air goes into our lungs. So now these lung cells will actually give out oxygen into the bloodstream so that all the cells can get oxygenated, and they will actually take in carbon dioxide to exhale it outside the body. So as you can see, lung cells are in a way different, but still the process of diffusion always holds. So next we'll look at the process of osmosis. So osmosis is the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane. In reference to cells, this semi-permeable membrane is the plasma membrane, as it allows the entry of some particles while it rejects the entry of others. So now this video clip shows the process of osmosis clearly. So as you can see that this is basically the membrane layer. Over here we have clear water. It has nothing in it, and as you can see over here, these are salt particles. So these are salt molecules dissolved in water. So what osmosis does is that this clear water is going to rush into this side. One might ask why, because over here there's large quantity of clear, or you can say pure water. Over here it's mainly contaminated, or it has salt diffused inside of it. So basically, so that the ratio of clear water is the same in both the areas, this clear water which was filled over here is going to move here. And now, as you can just see, this is reverse osmosis. It only happens if you apply pressure, because the normal state is it going into here from this is the region of high concentration of clear water molecules. This is a region of low concentration of clear water molecules, so it's going to move here. But if we apply pressure on this side, then reverse osmosis is going to happen. So then what will happen, that this dirty water is going to go inside here. Next up, we'll look at the type of solutions. So the first one being the hypotonic solution. So if the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water through osmosis and swell up. Such a solution is known as a hypotonic solution. So this image shows a red blood cell in a hypertonic solution. So basically, in a hypertonic solution, there's a large amount of water outside the cell than inside the cell. So high concentration outside, low concentration inside. So water will move into the cell. As a result, the cell is going to swell up. It's going to basically form a circular bubble. Next up is an isotonic solution. So if the medium has exactly the same water concentration as the cell, there will be no net movement across the cell membrane. In this type of solution, the cell will remain the same size. Such a solution is known as a isotonic solution. Now this is a red blood cell in an isotonic solution. So basically, if the water concentration over here is X, and then for an isotonic solution, the water concentration inside the cell is also X. So basically, nothing will go in, nothing will go out, and the cell will just remain the same size, same shape. So this is basically a healthy red blood cell. So our bloodstream, basically the water concentration in our bloodstream has to remain the same as the water concentration inside the red blood cell. That's why everyone says drink water, so that basically your cells don't die. The last is the hypertonic solution. So if the medium has a lower water concentration than the cell, meaning that it is very concentrated, the cell will lose water by osmosis and shrink. Such a solution is known as a hypertonic solution. So this is a red blood cell in a hypertonic solution. So outside there's lower water concentration, inside there's higher water concentration. So as the process of osmosis still follows, this high concentrated water will move out of the cell. As a result, the red blood cell will shrink in size. So now we'll look at the composition of the plasma membrane. So the plasma membrane is composed of lipids and proteins. Lipids are basically fats and proteins are bigger forms of amino acids. So this is a very simple way of how the plasma membrane looks under an electron microscope. So what this is the lipid bilayer, or basically it's composed of lipids, these fats, and these are just protein capsules. You don't have to know much about this in detail. It's just to let you know that this is how the plasma membrane is structured. And the last thing associated with the plasma membrane is a process known as endocytosis. So lipids and proteins allow the plasma membrane to be flexible. This flexibility allows the cell to digest or grab food from its environment. This process is known as endocytosis. So as you can see over here, there's a food particle entering the cell's vicinity. So what this cell is a amoeba. So what the cell will do, it will form this kind of pseudopodia, it's called. It's basically going to try to cover the cell or wrap the food particle around it, and then once it enters, it makes a food vacuole. So then see, as you can see, the pseudopodia basically encapsulates this food particle, and it's going to enter the cell, forming a food vacuole, and then all the enzymes are going to act on this food, and it's going to digest this food. Now, this whole process is known as endocytosis, and it's only possible due to this flexibility of the plasma membrane. So what we covered today, we covered that Robert Hooke discovered cells in the year 1665, that there's a division of labor in every living being, and that we covered the first main component of a cell, which is the plasma membrane. We looked at how it helps in diffusion, osmosis, and the three types of solutions, the hypertonic, isotonic, and hypotonic solution. And we in the end we looked at endocytosis. So that's going to be it for the day. Thank you, and we'll see you next time.